We can isolate the clones following pyrimycin selection and characterize them, and what you can see is that in each population, using PCR, we can actually validate that that gene only went into one site in the human genome of this cell line, and in either direction. There is a small background, but the specificity is over 98%, so if you collect 100 different cell lines and assay, you'll only have 2 cell lines that are derived from a background integration of that element. We've also optimized the relative efficiency by changing the number of zinc finger domains. And you can see that on this plate of cells here. As we reach about 4 zinc finger domains, we begin to plateau in terms of specificity. I'm going to move a little bit quicker here. The specificity, however, is quite high, bouncing around into the high 90s as we add these zinc finger domains. A 4-finger protein in this sense is actually binding a DNA element of 44 base pairs in length, because that gives us an address that can be quite specific within the human genome. These catalysts are orthogonal in that if we take a catalytic domain from one serine recombinase and challenge it with a site from another, that would be this sort of mixture of G and T shown at the bottom. Those domains don't communicate. That is just illustrating that there's a specificity that comes with the catalytic domain that we're exploiting in this approach. Now, in the last 1 minute and a half, what we've been focused on is trying to relax the specificity of that enzymatic domain, or to direct it in a rather rational way. And the idea here is that we would then be able to target any gene or any site in the genome if we had a generalist catalytic domain. So that catalytic domain ideally wouldn't care about this core sequence, and its specificity would be directed by just the zinc finger domains themselves. To solve this problem, we used applied molecular evolution, and this is the approach. We have a plasmid bearing a library of recombinase variants, introduce that into E. coli. E. coli makes the enzyme. If that enzyme is active, it acts back on these 2 recombination sites, excising a piece of DNA, leading us to isolate now using PCR the unique plasmid out of many billions that underwent this catalytic transformation. And what these DNA gels just simply illustrate here is that after 7 or 8 rounds of molecular evolution, this protein, which was dead, we're taking a protein and trying to make it do recombination on a site that it was not evolved to act on. We can reanimate it using molecular evolution now to act on that site. I won't go over any of the mutations. We can verify that indeed we've loosened the sequence specificity. So basically, any base pair is taken in that core region. Significantly, if we change the zinc fingers, the protein works only on those sites bearing the zinc fingers. So we've really mitigated the specificity of the catalytic domain and now can overwhelm that with the specificity of the zinc finger domains. And then finally, this is our first sort of experiment in endogenous targeting. This is where we took a site in the human genome at the ERBB2 site, made 2 unique zinc fingers, added our generalist catalytic domain, and asked if we could deliver plasmid directly at that site. And this is our first evidence that we can make directed delivery at a site in the human genome. Finally, to close, the basics of the technology, at least in terms of DNA binding, are now automated. You can go on the web, type in a DNA sequence, you can type in a promoter sequence even. You find all the sites that you can rapidly build proteins to bind to. We're still working out the fine details of how we think in the near term we'll be able to use these as tools to solve this gene therapy problem of actually site-specifically inserting a gene into any site within the human genome. And finally, I want to thank the individuals that have worked on this project. As I said, it was overall, I touched on work that took place over about 15 years, and these are students and postdocs that have passaged through the lab and worked on one aspect or another on this project. These are the individuals on the last section of the project concerning zinc finger recombinases. And finally, thank you.
Beautiful dog. Can you get so-called